the other things. See, we have got three different kinds of muscles are there. Cardiac muscle, skeletal muscle, smooth muscle. Right? Smooth muscle is there with all our internal organs, like blood vessels, internal organs like kidney, intestine, all of them has got smooth muscle. Skeletal muscle, the name it is given because it is attached to our skeleton. The bones are our skeleton. To that skeleton, a muscle is attached, that is called skeletal muscle. Third one is cardiac muscle. When you see the functioning, skeletal and cardiac muscle functioning is very similar. Smooth muscle has got a slightly different function. Leave that aside. But think about our skeletal muscle. What happens when you are continuously working? Let's say you are continuously running. What happens? You are using your skeletal muscles. When you continuously use, there is a chance to get spasms. You know what do you mean by spasms? Contraction, relaxation is a normal process. When muscle contracts and stays as such, it is called as spasm. Usually, night times when there is a deficiency of potassium, people will get calf cramps. Calf is this area. At that area, muscle undergoes spasm. That is called as calf cramp. Understand the concept. Skeletal muscles, when they are overly used, there is a chance of getting into spasm. So they will be in contractile state. That's it. They will not get relaxed. What happens with cardiac cell? Now see, skeletal muscles, when they are overly used, they will undergo spasm. What is happening with cardiac muscle? Cardiac muscle is always functioning. Before our birth itself, in the mother womb itself, heart rate is there. In fact, in mother womb, the heart rate is more than what we have. It is 140 to 180. It is more than double. But when we come out, it stabilizes and becomes 70. All the time you sleep, you do work, you don't do work, whatever you do, heart has to do its work. Continuous contraction. So continuously working. Does spasm occurs to cardiac muscle? If occurs, what happens? If spasm occurs, heart will stay as such. And this is called as cardiac arrest and people will die with them. Spasms do not occur to cardiac cells. Why spasm is not occurring? It is because of a special arrangement in sodium channel. There is something called as refractory period. Refractory period means, see, the action potential begins with sodium entry. Why? Because sodium channel is opening. Now after that, immediately in milliseconds, sodium channel closes. After that, lot of channels are opening, closing, and all these things are happening. Until the entire cycle is completed, again the sodium channel will not open. If it opens here or here, you will get one more cycle. That is the problematic one. That is what happens in certain kinds of arrhythmic conditions, diseased conditions. All the populations, the sodium channel will be refractive. Once it is closed, it takes at least this much time again to get open. Because there is a special arrangement in sodium channel. So there is a uniqueness in this sodium channels. That sodium channel, now sodium channel has got three forms, three isoforms. The forms are, one is known as closed in rest form, and then open, and then closed inactive form. Closed inactive form. So the receptor will be in three forms. Initially, the sodium channel is in closed form. Whenever there is a potassium change occurs, the closed form moves into open form. Open form is nothing but the channel opens. Once the channel is open, sodium rushes inside the cell. After that, immediately it goes to this closed but inactive form. That means after that, immediately the channel closes. But in order to get open, it will not immediately get open. It has to undergo to this closed rest form, then only again opening. So this period is known as refractory period. Refractory period means unless the closed inactive is converted to closed resting form, it cannot open again. So it takes some time for this closed inactive to convert into closed rest form. During that time, this entire process takes place. That means in between, you don't see opening of sodium channels. Why? Once it is open, it goes to this inactive form. It will not immediately open. No, it has to go to this closed rest form, then it has to get open. This period is called as refractory period, and in this meantime, you don't see one more sodium channel open. If sodium channel is not open, all these changes will not occur. One more calcium burst will not be there. If this is happening, you don't see that spasms. Why spasm occur? when there is excess amount of calcium, it causes continuous contraction, and that is what results in spasm. So if per one cycle, certain amount of calcium comes, that's it. It is not continuous burst of calcium. Why it is not continuously coming? Because sodium channel is regulating the levels of calcium. How? Once it is open, it goes to an inactive form, then only it is converted to resting form. After that, it opens. During this time, the rest of action potential events takes place, and that is what becomes a refractory period. If this process is not there, cardiac muscles could not survive for long, because all the time it is working. In fact, we work lot and lot because of anxieties and stress. It will be working harder and harder, but still it doesn't undergo spasm. If it goes, we all die. It happens only in certain conditions, in diseased conditions. In certain people, there is a problem with sodium channel. It is called as channelopathy. See, pathy here indicates not the husband. Pathy means pathology. The word is derived from pathology. Channelopathy means there is a disease, there is a problem in that sodium channel. Because of that problem, it is continuously releasing sodiums. At that case, one more action potential comes here, and that results in arrhythmias, continuous contraction, some problem with calcium and everything. In normal population, it is controlled. The refractory period is because of this arrangement of sodium channel. What happens is, usually sodium channels are in closed form. That means the channel is closed. Once the voltage comes, channel opens, sodium gets inside. Now, after that, again, it goes to closed form, but the closed inactive form, again, it has to go to resting form, then only it will get open. And during this period, all these events will take place. So immediate opening of sodium will not take place. In order to get open, it has to move to this form, and then only opening. And this will save from spasms. So this is the important things in electrophysiology. Let me repeat this again. See, till now what all the things we have seen is, cardiac cell physiology is based on its ability to cause contractions. What is the function of heart? It is a muscular pump. It is giving contractions. During the contractions, blood will be going to lungs, or it is going to all the parts of the body. That is what is happening. And this function is carried out when heart can give these contractions. How and why it is giving contractions? It is because of action potentials. Now, what is happening in this action potential? There is something called as resting membrane potential. All the cells has got a negative potential inside the cell. In case of heart, it is minus 90 millivolt. When the potential is changed, when the voltage is changed, again see the voltage change starts from SAnode. Hence, they are known as pacemaker cells. The voltage change starts from that SAnode. From there, the voltage change is transferred to all other cells. One more thing. Cardiac cells has got a special property. The cells are called as intercalated cells, or intercalated discs are there. It is also known as gap junction. See, normally every cell has got cell membranes around it. Cells will be like this, right? The continuous cells will be having this cell membranes, and they will be continuously building tissues like this. In case of cardiac cells, there are certain gaps between the cells. That means if the potential is changed here, it can be transferred to the other cell. From here, the potential can be transferred to this gap junction. There is a gap in the junction, hence it is known as gap junction. Again, the potential can travel to the adjacent cell. This is what happens in heart. When you see the heart is like this. So the signal generates here, the entire atria will undergo contraction. Why entire area? Because all the cells are connected. One change in potential will be transferred to other cell, and all the changes occurs in all the cells. Again, same thing. When AVnode gives that signal, the entire atria will undergo contractions. Why? The cells are not separated. They are connected by gap junctions. That means potential can be transferred from one cell to another cell. Once the potential change is there, what happens? All these potential or voltage gated channels will open. The entire phenomena takes place. The important one is phase 2, wherein calcium gets inside and causes contraction. The other one, when one cycle is taking place, sodium channel opening will not occur, because sodium channel has got a special arrangement. Sodium channels will exist in 3 forms. Initially, it is in closed form. When the potential change is there or voltage change is there, it goes to open form. After that, it gets into an inactive form. Then only it will be converted to closed form. During this time, the remaining electrophysiological events takes place. Now, what all the things I have explained, it is about this entire cardiac muscles, leaving these nodes. Nodes are the one which is generating this signal, and they have this automaticity. Remaining cells will take the signal from that nodes, and because with the help of these gap junctions, the entire cells will undergo a coordinated muscle contractions. Now let us see what happens in nodal tissue. See, nodal tissue's ability is to generate spontaneous signal generation. Spontaneous means without any neuronal supply, they can generate their own signal. That is what is called as automaticity. Now, sometimes what happens is the SCN node is the pacemaker cell, but sometimes SCN node function can be carried by adjacent cells. When this happens, it results in ectopic pacemaker generation. Ectopic means out of the place. It is also results in ectopic beats. We'll see one by one. Now see this. The only difference in SCN node and AV node is they have got a special type of calcium channels. The job of the channel is, see what is happening here in this normal remaining cardiac cell electrophysiology, calcium opens at this voltage gated channel and then closes out. But when you see the action potential in SCN node, it looks like this. You can see there is a continuous rise of potential. Again, potential generates, peaks come down and like this. So there is not a flat line continuously. Again, it is rising. Again, the potential rise is there. Why potential is rising? Because at this junction, calcium gets into the cells. There are special calcium channels in SCN node and AV node, which will continue to release. And because calcium is getting inside the cell, again, the potential rises. When it is rises, again, it gives an action potential. Everything happens. Again, it comes down. So at these junctions, calcium is causing action potential. In normal cells, the action potential rises because of sodium entry. Whereas in case of nodal cells, nodal or automaticity cells. In these cells, the potential rise is because of calcium. Remaining all things are similar. So this potential rise occurs because of calcium. At this junction, calcium closes down. Again, potassium is going down, going out. Again, a plant phase is there. Again, repolarization occurs, and then again, all these changes will. So the reason why nodal cell shows their automaticity is they have got a special type of calcium channels which will continue to leak into the cell. When calcium is coming inside, what is the charge of calcium? Again, it is positive. When there is a positive, what happens? Depolarization. So again, rise of depolarization is there, fall is there. So the entire cycle, this action potential, all these things will go on. And this signaling continues to go on. That is the reason why they continue to give the signals. When they are continuously giving signals, the signal will be spread to all the cells and every cell respond with this manner. Let me repeat it again. Understand this word. See what is happening here at this junction, the polarity is changing. It is known as excuse me, depolarization phase. Instead of negative, it is becoming positive. After that, what is happening? Again, polarity is becoming negative. This phase is called as repolarization. What is polarity? Negative inside. What is depolarization? Turning it to positive one. What is repolarization? Bringing back into normal polarity. So there is a difference. If this is the baseline, if it is going down further, it is called as hyperpolarization. If it is coming to the normal level, it is known as repolarization. If it is going to positive direction, it is known as depolarization. Simple words, D opposite, re bring it back. Going further down, hyperpolarization. But this is what happens in HCA and AV nodes. HCA and AV node. This is how the nodal cells will continue to give contractions. That is why HCA node is called as a pacemaker cell. It starts the pace. goes to each and every cell, and then it goes to AV node, and then it continues to grow. So the differences are, see, majority of cardiac muscle is made up of these cells. You have only few spots for the SA node and AV node. Their job is to give continuous signals. How they are giving continuous signals? Because of a special type of calcium channel which is releasing calcium. Because calcium has got a positive charge, again the potential rises, and that continues to go on. So continuously they will be giving signals. The signals will be taken by the adjacent cells. They will respond accordingly. Calcium get inside, signal continues. So in cardiac physiology, you need to keep these things in mind. The non-automatic cells action potential and the nodal cell or automaticity containing cells action potential. Action potential is simply rise and fall of this depolarization. Now the last thing. We need to see how the calcium is exerting its effect. I keep saying that whenever calcium comes inside the cell, it causes the combination of actin and myosin, and that is what results in contraction, cardiac cell contraction. Again, let me tell you, skeletal muscle and cardiac muscle will show similar patterns of contraction function. Smooth muscle has got a different thing. Now let us understand how this calcium is causing this kind of contraction. Now before getting into that, understand certain things. See, calcium, we have seen sodium and potassium ionic equilibrium is set normal because of this sodium-potassium bond. But what happens with calcium? Calcium also coming inside the cell. Any slight increase in calcium will immediately may cause contractions. So the concentration of calcium inside a cell is very tightly regulated. So during potentials, calcium is coming inside the cell. I explained about what happens with sodium and potassium. Their concentration is corrected with the help of sodium-potassium bond. But what happens with calcium? So whenever calcium gets inside the cell, inside there is an organelle which takes up that calcium. That is called as Sarcoplasmic Reticulum, SR. The job of Sarcoplasmic Reticulum is whenever calcium gets inside, it exerts its action. After that, the calcium is taken back into the Sarcoplasmic Reticulum. Once calcium gets inside the Sarcoplasmic Reticulum, the free availability of calcium will not be there, further contraction will not happen. That is the reason why cardiac cell will undergo contraction. After that, calcium is taken back by Sarcoplasmic Reticulum. It gives relaxation. Now, it is not completely taking that calcium. 70% of intracellular calcium gets inside the Sarcoplasmic Reticulum. Rest of 30% is taken outside the cell with the help of calcium ATPase and sodium calcium exchanger. Three things are there. During action potential, you have ionic disturbances are there. Sodium is coming inside, potassium is going outside, and calcium also coming inside. Sodium potassium ionic concentration is readjusted with the help of sodium potassium bond. What it does, it pushes sodium outside the cell, takes back potassium. So sodium potassium levels are equilibrated. What happens with calcium? After the contraction, the calcium, the free available calcium is taken back into Sarcoplasmic Reticulum. Whatever calcium is coming inside, 70% is taken back into Sarcoplasmic Reticulum. Rest of 30% is sent outside the cell with the help of pumps. Again, one pump is there called as calcium ATPase. So the calcium is sent outside the cell with the help of calcium ATPase, or there is an exchanger, sodium calcium exchanger. That means calcium goes outside the cell and sodium comes inside the cell. This is called as sodium calcium exchanger, NCX. We'll see the details now. Now see, imagine this one is a cardiac cell. There is a special arrangement in cardiac cell. It is known as T-tubule, cardiac as well as skeletal muscle. Now, when potential change occurs, the T-tubule contains calcium channels. So when there is a potential change, as we have seen here, the calcium gets inside the cell, Ca+2. These channels are known as L-type calcium channels. You know, there are so many types of calcium channels are there. Very prominent one are L-type calcium channels. Now, these L-type calcium channels are present in cardiac tissue as well as skeletal muscle. So when there is a potential change occurs, this is cell membrane. See, cell membrane This is a kind of inverted T shape. See the T, inverted T shape, hence it is known as T tubule. Common. Now, at this cell membrane, when the potential is changed, this potential change is there when it is converting into positive, the voltage gated calcium channel opens. That particular calcium channel is known as L-type calcium channel. When the channel opens, calcium gets inside the cell. Once calcium gets inside the cell, see, I told you about sarcoplasmic reticulum. Sarcoplasmic reticulum is the one. This is sarcoplasmic reticulum, which contains a lot of calcium stores. There are so many calciums are present inside the sarcoplasmic reticulum. But sarcoplasmic reticulum has got a special type of receptor known as Rhianodine Receptor, RYR. Rhianodine Receptor. Now the job of this Rhianodine Receptor is it will not allow calcium to go into the cell. So this is a complete cardiac cell. So it will not allow the calcium getting into the cell. But when calcium channel is opened, when calcium comes, this calcium acts on this Rhianodine receptor, and because of that effect, calcium comes up. So the entry of calcium makes sure from the sarcoplasmic reticulum, calcium is coming into the cell. Now, once calcium levels are increased inside the cardiac cell, they will be combined with actin. Now, actin myosin combination results in muscle contraction. But on the actin, the binding sites of myosin are blocked by tropomyosin. There are two protein fragments out there, troponin C, tropomyosin. The job of tropomyosin is it will block the binding sites of myosin on actin. Imagine this one is actin muscle fiber, and to that myosin will bind here, and the sites are blocked by tropomyosin. Now, the job of calcium is whenever calcium comes, the calcium will changes the conformation of this tropomyosin and removes that block. Then actin myosin combination occurs, and this is what results in contraction. So this is the job of calcium. I'll repeat this. Whenever there is an voltage change is there, the L-type calcium channel opens. Because of that, calcium gets inside the cell. The calcium will activate Rhianodine receptors on the sarcoplasmic reticulum. So sarcoplasmic reticulum, once this Rhianodine inhibition is removed, calcium is released into the cell. Once it is into the cell, the calcium binds on the actin. You have tropomyosin, troponin C is there. It binds with them, removes that inhibition, and actin combines with myosin. Now this is what results in contraction. Now, after that, you have a lot of amount of calcium is present inside the cell, which is not good. It has to be taken back. How it is taken back? It is taken back by a particular pump known as sarcoplasmic endoplasmic reticulum ATPase. Calcium ATPase. It is known as SERCA. Sarcoplasmic endoplasmic reticulum calcium ATPase. The job of this one is calcium, with the help of this one, again gets inside the cell. Whatever the calcium is released, once it causes contraction, it is immediately taken back into sarcoplasmic reticulum with the help of this particular ATPase. 70% of the calcium gets back into sarcoplasmic reticulum. Once sarcoplasmic reticulum gets this calcium, free availability of calcium will not be there. Again, active sites which will bind with myosin are covered by tropomyosin. Combination will not occur, contraction will not be there. 70% are removed like this, but remaining thing also removed by a calcium ATPase pump. The job of this calcium ATPase is calcium will be taken outside of the cell with this pump. Now there is one more pump is there. There is one more pump is there. It is called as sodium calcium exchanger. How it works? It sends calcium outside the cell. At the same time, it brings back sodium into the cell. So 3 of the sodium brings back into the cell. 1 calcium goes out of the cell. So finally, what is happening? Calcium is getting removed out of the cell. Calcium is removing out of the cell. Calcium is getting inside sarcoplasmic reticulum. When calcium levels falls down, further contraction will not happen. This is what happens. This is known as sodium calcium exchanger, NCX. Remember, sodium calcium exchanger works with the help of sodium potassium pump. How sodium potassium pump acts? It sends free sodium out of the cell and brings back 2 potassium into the cell. And these 3 sodium taken back into the cell with the exchange of calcium. So in order to remove this calcium, you need the pumping ability of sodium potassium pump. You know, digitalis glycosides acts like this. They inhibit this pump, calcium is not removed, calcium levels increases, causes contraction. We'll see in the later classes too, but understand how the contraction, how the calcium effects are on cardiac muscle cell. On the cardiac muscle cell, this is how calcium contraction occurs. Let me repeat this. There is a special arrangement known as a T tubule. It is nothing but invagination of cell membrane is there. So when the potential change comes, because of voltage change, L-type calcium channel opens. That allows calcium into the cardiac myocyte. Once it gets inside, it acts on Rhianodine Receptor, which is present on Sarcoplasmic Reticulum. That enables Sarcoplasmic Reticulum to release calcium. Once calcium is released, the total calcium ion concentration increases. They will act on actin troponin C. Troponin C, tropomyosin will block the binding sites of actin with myosin. When calcium changes the conformation, actin will be combining with myosin, and that results in contraction. After that, calcium is taken back again into sarcoplasmic reticulum. With the help of a sarcoplasmic endoplasmic reticulum calcium ATPase. This is a pump. So whenever you read about a pump, that indicates it is using energy. You know, we all use pumps to get the water from wells. Unless you use that pump, you cannot get the water back. You are using energy. Similarly, this is using energy. Again, when energy is required to pump against the concentration gradient, we take the water against the gravity. If you are pouring water, it just goes down according to gravity. If you are moving against the gravity, you need to use a pump. Similarly, against the already calcium is more inside the sarcoplasmic reticulum. Again, you are sending to higher concentration of calcium, hence it is known as a pump. After that, there is a calcium ATPase. Again, it sends calcium to the outside of the cell. One more thing is sodium calcium exchanger. It exchanges 3 sodium with 1 calcium. And this pump works well when sodium potassium pump is working. So this is what is electrophysiology of cardiac cell. Now see, I have explained some physicochemical and biophysical properties of cardiac cell. Understanding action potential, understanding the chain of potential is very important. When we read about arrhythmia, all arrhythmic drugs will act on this cycle because there occurs the changes in this action potential. That is what results in arrhythmia. Now, SAnodes, because of this calcium channel, they have this particular property, and that is what is causing them act as pacemaker cells. And how the contraction everything occurs, this is how it occurs. Contraction is not permanent. Immediately calcium ions are taken back, hence relaxation occurs. So today we have seen what is membrane potential is, what is resting membrane potential is, and then how the action potential occurs in non-automatic cells, and then how the action potential occurs in SAnode and AVnode, and then how the refractory period is occurring by using sodium channels. And we have seen the special function of sodium potassium pump. Finally, we have seen how the contraction occurs in a cardiac cell. This is very similar to skeletal muscle too. A slight difference is there. But smooth muscle, there is a different thing is there. Here you have troponin and tropomyosin. There you will be having calmodulin. That is the difference. We'll see when I discuss it there. This is how it happens. One more important thing. You know, last class I have explained about beta receptors. What is the function of beta receptors? They act through a GS subtype of G protein coupled receptors. What happens with GS subtype? GS subtype increases adenylate cyclase enzyme. If adenylate cyclase is increased, what happens? Cyclic AMP increases. That is the second messenger. What is the function of cyclic AMP? It activates protein kinase A. Now comes the job of protein kinase A. Now see, when beta receptors are activated, protein kinase A is activated. Kinase means it phosphorylates. What it phosphorylates? It phosphorylates calcium channels. When calcium channel is phosphorylated, it opens up. More calcium gets inside. It phosphorylates rhinodine receptor. When rhinodine receptor is phosphorylated, it releases a lot of calcium. It phosphorylates this pump. It also phosphorylates a troponin inhibitor. The job of this troponin inhibitor is it is inhibiting not to occur muscle contraction. It is blocking the muscle contraction. So this is how protein kinase A acts. It is phosphorylating calcium channel, more calcium get inside. It phosphorylates rhinodine receptor, more calcium get outside. It removes that inhibitory factor and troponin so that activation occurs. All these effects increases calcium. That is the reason why beta receptor causes positive inotropy, positive chronotropy, positive dromotropy, positive bathmotropy. This is the biochemical basis. This is how beta receptors act. So this is about cardiac physiology. You will be provided notes.